Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Good morning one and all. Now I'm now ready with the topic agarose gel electrophoresis. In the previous class we have already discussed about the separation of proteins by SDS page. Okay, now let's start with the separation of agarose gel electrophoresis. So you have already deal with SDS, right? So now let's go with agarose. What is agarose? Agarose is a derivative of agar. Okay, so based on the percentage of the agarose used for the lab for the DNA, they take the percentage of agarose. Generally, if the molecular if percentage of gel is percentage of agarose is used less, you can get higher molecular weight of the DNA. Okay, it is always like that. Okay, so for example, agarose is the percentage is used for agarose for gel is percentage less. You look at the larger fragments of DNA separated. In the same way, generally the percentage of agarose, like 0.7 to 1% of agarose is gels are used for the DNA separation. Okay, so now let's start how agarose gel electrophoresis moves. So the main difference is the difference between the two electrodes is very important. Yes, here what happens is you all know that anions move towards your anode and the cations move towards the cathode, right? So anions are negatively charged particles, they move towards the anode, and cations are the positively charged particles, move towards the cathode. Okay, so when the electric field is placed, or when electric field is placed, when the gel is running, the main thing depends on the potential difference between the electrodes and the distance between the electrodes. Here I want to show you one thing. See, this is the thing that I'm talking about. Suppose these are the two electrodes that are present here. Okay, the distance between the two electrodes are 10 centimeters. Okay, and the voltage that you have applied for is like 100 volts. Let's assume the voltage that you have given in this example is 100 volts, and the resistance between the two electrodes is 10 centimeters. Okay, now the electric field is always dependent on the distance between the electrodes and amount of voltage that you have given. So that is a formula, right? That just now we have gone through that. So the distance amount of the amount of current you have been supplying divided by the electrodes. So if it is like 100 volts, out of that the 10 centimeters is the distance, so the electric field you have to apply is 10 volts per centimeter. The more the distance between the electrodes, less time it will take for the process to get completed. So this is the main basic principle for agarose gel electrophoresis. Okay, see, and in agarose gel electrophoresis, what really happens? Agarose is taken, agarose is added with a buffer. The buffer that is used for agarose gel electrophoresis is TAE buffer. What happens is actually is it's a chelating agent. Sorry, it's a chelating agent. What happens is when you want DNA in a native form, okay, this particular DNA native form you want it, right? So this particular EDTA would reduce the action of magnesium ions, which acts as a catalytic agent for the DNA nuclease enzyme. DNA nuclease would denature your DNA, but we want that in a native form. In order to reduce the activity of that DNA nuclease, you will add this particular EDTA to that buffer. That would reduce the magnesium ions which are responsible for the DNA nucleus to function. So that is how DDTA is important for the buffer. Okay, and next comes what is the when you add that particular EDTA buffer to the sample, and it should be immediately well mixed. So for that mix you have to, it has to get mixed with that agarose. So you have to immediately place it in a microwave, and then immediately you should place it in the water bath for further cooling, right? It should come around 55 degrees. Once it is reached to that temperature, it is ready for the gel to be casted. So in meanwhile, you can make a gel out of it. Like you can just form a small with a comb and all. You can just place it, and once the gel tank is ready, just pour the gel and let it get solidified at room temperature. Just place the comb immediately and leave it as such for the walls to be well framed. Okay, once the whole setup is made, you are ready with your sample, and you also be ready with your gel. Once the gel is solidified, remove all the things along with the comb also. So now place it in the buffer tank. Okay, now place it in the buffer tank. You have your sample ready of that. Okay, in that sample you better also add so I also told you importance of ethidium bromide. What does ethidium bromide is? Ethidium bromide is added into the sample before you load it into the gel, because ethidium bromide is a mutagen and it is very dangerous thing. You have to use it under a very important precautionary conditions. Okay, what does ethidium bromide does? It is an intercalating agent. It would only bind to the DNA bases, the nitrogen bases that you see in the DNA, so that when you place this particular sample in UV light, you should be able to see the DNA bands clearly. More the DNA present in that particular sample, you will be getting the visibility under UV light. Okay, so this ethidium bromide acts as an intercalating agent. It would directly bind to the DNA bases of the DNA for further visualization. This should be added in the sample before you load it. Once you load the sample, the first well should be loaded with a DNA ladder of known molecular weight. So for example, it should be like, see, based on the base space, DNA is measured in base space, right? So based on the higher base space goes in the top to be in the top, and lower base space will move faster and get settled at the bottom. So if you want a comparison, you have to add the DNA standard ladder as a marker at the first well. Then all the three samples of to be which are to be separated have to be added immediately to the remaining wells. So once the current is passed, you will get to know the slowly all will move at a time, and once the whole bands have moved down, you can switch it off. Once it is cooled down, you can take it out and place that particular agarose gel into a biorad for further analysis. So they will get to know, there you will get to know the bands how they are. See, this is the sample that I was talking about. Okay, this is where you load the marker. This is where you sample 1 and sample 2. Upon the passage of current, this moves towards your down. Okay, the direction of this is the buffer that I have told you. TAE and TBE buffers are mostly used for DNA and RNA separation. Okay, so next comes how the understanding of mechanism of DNA is. So this is the length of the DNA fragment, the 1000 base pairs at the top and 100 base pairs at the bottom. So this is the marker of known size. Okay, sample A, sample B, sample C, based on the size of size, they will be dependent. For example, if you got a sample of complete you got a certain 100 strands of 1000 base pairs and 50 strands of 900 base pairs, all the 1000 strands will be of sample A will be here only. Okay, it's not one strand. Okay, all the strands of same molecular weight will come under this base or under this band. Okay, if 900 base pair, you've got 5 to 10 base pairs of 5 to 10 fragments of 900 base pairs, then they will get stopped here. Okay, this is how you have to understand the mechanism. So based on it, we got a graph where the amount of distance moved reduces the molecular size. More the distance moved, lesser the molecular size becomes. So okay, the highest molecular size will have a very zero distance moved. Okay, that is a relationship between the fragment size and the distance moved by the fragment. So hope you got an idea about agarose gel and SDS page regarding electrophoresis. Now in the next class I will talk about the chromatography techniques, how the separation happens. So I am dealing only with the basic principle and how in the lab condition. Hope you understood the class very well. So see you in the next class. Please subscribe my channel, like my channel, and comment me in the comment box. Okay, and have a nice day. Bye.